Bio 181, chapter 15. And we're talking about the central dogma and creating proteins. And the second step is called translation. Translation is converting messenger RNA into a protein. So we're taking the information that messenger RNA has copied from DNA, and now we're going to make a protein. We are going to require ribosomes and tRNA for that process. So just a little overview here. So if we have, here's my nucleus. Let me draw my cell. Okay, so here's a cell. And we have a eukaryotic cell. And the nucleus, we've got our DNA, double helix, that is transferred into messenger RNA. Messenger RNA is then modified, spliced together, shortened up. We put a 5' cap on it, a poly A tail, take out the introns, and it's ready to go. Then the messenger RNA, which is this, okay, then leaves the nucleus, and this is where we're at. We're in the process of translation. So previously in the nucleus, this was transcription. Transcription, right? This process here. And now out here, we're going to do translation. So translation. Okay, so translation. Messenger RNA is coming out. I'll just do it in blue here. So here's my messenger RNA. It's single-stranded. It leaves through these holes in the nuclear envelope. Okay? And it's going to go out into the cytoplasm and find a ribosome. So ribosomes consist of two subunits. They have a large subunit and a small subunit. It's kind of like a hot dog bun. It's going to sandwich right on the messenger RNA. And it's going to read off the letters on the messenger RNA. And as it does so, it's going to read a sequence here. So let's see, it's going to read a sequence. Maybe the sequence is UCG, and I've just made that up, so I don't really know what that is. So it's a sequence right here. That's going to correspond to a specific amino acid. So there's an amino acid. And remember, an amino acid, this is not to scale, this is much larger, is the building block of a protein. It's the monomer of a protein. So this is going to specifically correspond to a given amino acid, and then that amino acid is going to come into the ribosome. So that's where the transfer RNA comes in. So the transfer RNA is going to be a sequence that's made out of ribonucleic acid, and it's going to be attached to an amino acid. It's going to then bring that into the ribosome, and it's going to sit kind of right in here. There's a little place for it to sit. It's going to bring in that amino acid. Oops. And it's going to start the chain of a growing polypeptide or protein. Then we're going to read the next sequence here. 1, 2, 3. There's going to be another different amino acid. Maybe I'll make it a similar color, right? So here's another amino acid. It's another one. And that's going to be brought in by another transfer RNA that's hanging out near the ribosome. Okay, it's going to bring that one in, and it's then going to be bonded to the amino acid that was previously there. Ultimately, we're going to get a whole sequence of amino acids, and that's going to be our protein. So the 3-letter sequence, the genetic code, is read in a 3-letter sequence called a codon. So what was the previous one? I'm just curious now. So I said UCG. So if we look up, so the first letter is U, the second letter here is C, the third letter is G, okay? So it turns out that's cysteine. So the amino acid that corresponds to that three-letter sequence is cysteine. And you're going to have, since we have a total of 4 different letters possible, okay? There's 4 times 4 raised to the power. It's going to give us 64 different possible codons. So there's 64 codons. Now we don't have that many amino acids. We have about 20 amino acids. So that means we're going to have redundancy where for a given amino acid, like here, there's 2 different codons that are going to specify it. Similarly for cysteine, we've got U, this is really hard to see, but there's letters here. So UGU and UGC, those are both going to specify cysteine. So if that's read off from the messenger RNA, the transfer RNA is going to bring that in. So, oh, and that's called the generacy. So this is just what I just said. So there's multiple different 3-letter sequences that can code for the same amino acid. So isoleucine is another amino acid. Okay, so we've got a few steps here, keeping it really simple. Translation involves matching the transfer RNA codon with the messenger RNA codon. So we're going to match it up. We're going to form the peptide bond. And this peptide bond is just the name for the covalent bond. So they're going to share electrons. We're bonding adjacent amino acids together. And then we're going to move the ribosome. Okay. So I don't know. I'm going to see if I can do this simply. So I've got the transfer RNA. I'm going to draw it. Here's my transfer RNA. It's got an amino acid hooked onto it. Keep it a different color just so we know this is an amino acid. Okay. And it has an anticodon. So it's got a sequence at the bottom. It's folded over. So we could unwind this messenger RNA here. So this is RNA, right? It's single-stranded. It's made up of nucleotides. tRNA, where are we at? This tRNA, right, stands for transfer RNA, or tRNA. And if you wind this up, right, over itself like this, then it kind of has this cloverleaf shape that I'm representing here. So that's what I'm drawing. And at the bottom of it, there's a sequence. This whole thing is made up of RNA, but at the bottom, there's a sequence called an anticodon, and it's just the complementary pairing for the codon. So if we have messenger RNA here, and let's just stick with what I had. So I said, what do we say? UCG, right? So UCG. So we have UCG here. That's going to correspond to cysteine. And that means that on the anticodon, we will have, so U is going to correspond to A, C is going to correspond to G, and G is going to correspond to C. So the first thing that happens, so this is step 1, we pair these up, okay? It's going to bring in the amino acid. Remember, we've got a big old ribosome here that's kind of sitting around this area. Here's my small subunit, okay? I've got a big top of the hamburger bun here. That's my large subunit. Okay, this is my ribosome. Okay. And as the transfer RNA comes in, the amino acid's going to bond to the other amino acids that were there. So say I have a sequence that's already been growing, okay? Once the transfer RNA comes in, it's going to add on the next amino acid in the sequence. And then the ribosome is going to scoot on down and read off the next codon. So the next sequence, whatever that might be, maybe it's CCG, right? And then it's going to go find the transfer RNA that has the anticodon for that. And it's going to bring in whatever amino acid that corresponds to. It's going to attach it to the growing chain, and then the ribosome is going to keep moving down. So that's how we make proteins. So messenger RNA, that's the type of RNA that takes the information from DNA. Remember, DNA goes to messenger RNA, goes to protein. So it's taking the information, carrying it from the DNA to the protein. The transfer RNA is what's bringing, right? Brings the amino acids to the ribosomes, And then the last part, these are just for completeness, there's a third type of RNA. It's called ribosomal RNA, and it's what the ribosome is made out of. In addition to some proteins. So if we were to zoom in here, we'd see that there's these different parts of the ribosome, and it's made out of rRNA and some other proteins. So makes the ribosome, right? It's used to build the ribosome. What it's made out of. So what are you going to be asked to do on the exam? So given a sequence of DNA, so say I get a sequence like CCG, ACC, TAA, just making up something, right? So this is my DNA. You are going to need to then provide the messenger RNA for that. So you'll need to provide the messenger RNA. Whoops, I want to block myself space. So the C is going to pair with a G, C, right? The A is going to go with U, G, and A, U. Okay, so this is in the video on transcription. So that's transcription. And then for translation, we're going to take this information and make amino acids out of it. So every 3-letter sequence is going to be an amino acid. The other possibility is we could have a stop codon. There's 3 possibilities that say stop, and then there's one that's a start codon that's always going to be at the beginning, which is met. So to figure out what it is, so we've got G, C. So this is for messenger RNA. So I'm going to look for G on the first letter, second letter across the top here, G, okay, and then C. Third letter, GGC, yeah, down here. So GGC, so this is glycine. And so this is going to be GLY. Okay, I'm going to abbreviate it like that. And then I do the same thing for the next one. So UGG, so I get a U, G, where is it? UGG. Here, tryptophan. Okay, this is the one famous. Turkey's made this famous. It helps you to sleep. Tryptophan. So TR. I'm not going to spell it all out. So tryptophan. So TRY. And then the last one, just for completeness, to finish it up, Okay, let's do a fun color, purple. So AUU, A, and then U, and then U, A, U, my at U. Okay, so this is isoleucine. So isoleucine. That would be our amino acids. So these are the three when I was drawing it earlier. These are coming in from the transfer RNA. They're the amino acids. There's 20 different types, right? Transfer RNA is bringing it in. You're forming a peptide bond between them. So this bond, it's a covalent bond. It's called a peptide bond. Right? And then this growing chain, right? This is the protein that's being made. And another word for protein that is polypeptide. Polypeptide. Okay. Some proteins have multiple polypeptides in their fourth level or quaternary structure. Okay. So just to mention for prokaryotes how it's different than eukaryotes. Notice that in the prokaryote, there's no nucleus. In the eukaryote over here, the big distinguishing factor, what defines a eukaryote as a eukaryote, is the presence of the nucleus. So here's my nucleus. It's just showing that there's pores in it, which is how stuff gets in and out, but this is the nucleus. So in the nucleus, eukaryotic cell, we talked about that's where you're going to find transcription. And then translation is going to be found out in the cytoplasm. So they're separated in space and time. Whereas in a prokaryote, so over here, you've got them all occurring basically continuously. So you have your DNA, which is the blue circular DNA here. Here's my DNA. The process of transcription, you're taking the DNA and making the messenger RNA. So the messenger RNA in this picture is shown in pink. I don't have that, but basically it's this thing, right? That's my mRNA. And as soon as it's made, these little ribosomes are hopping on. So the ribosomes are these here. So this is the small subunit and the large subunit of the ribosome. So that's what's shown here. You've got multiple ribosomes on the same strand of messenger RNA, and they're creating proteins. So the proteins that are being created over here, right? Here's the protein, okay? All right. So that happens in the same place in the cytoplasm, pretty much in the same time. That's it.